Assalamu alaikum. In this video we are going to talk about T cell receptors and superantigens and the effect of superantigens on the T cells. First we will look at the T cell receptors. That what are the T cell receptors? T cell receptors are the receptors that are present on the T cells and they are specific for a specific antigen. T cell receptors are made up of two polypeptide chains. One is the alpha chain and other is the beta chain, and these chains are associated with the CD3 protein, and we know that the CD3 protein is involved in the signal transduction pathway. So there are some similarities between the immunoglobulins and the T cell receptor proteins. The immunoglobulins are made up of different segments, which includes the variable, diversity, joining and constant regions, and in the same way the T cell receptor proteins are also made up of these different segments. These different segments, which include the variable, diversity, joining and constant regions or segments, these segments are encoded by different genes, and these genes are present on different locations on the chromosome or DNA. You can simply say that these genes are present on different locations on the DNA, and during DNA rearrangement, these genes move from one place to another place, or their segments exchange material with different genes, and due to this DNA rearrangement we get diversity in the T cell receptors. So a specific T cell receptor will be specific for a specific antigen. All the T cell receptors are not same. All T cell receptors are different from each other due to the DNA rearrangement, and because of this DNA rearrangement we get a huge number of T cell or T cell receptor proteins, and this number is 10 to power 7. So we talked about that the T cell receptor is specific for a specific antigen, and when a T cell is activated, when a specific antigen binds with a specific T cell receptor, that T cell is activated and that T cell makes clones of it. That specific T cell will multiply in number to produce more specific T cells for that specific antigen. But there are also some differences between the immunoglobulins and the T cell receptors. The first difference is that the T cell receptor is made up of two polypeptide chains, but the immunoglobulin is made up of 4 polypeptide chains or 4 chains. 2 of the chains are called as light chains and 2 chains are called as heavy chains. The other difference is that the T cell receptor requires the antigen to be conjugated with the MHC protein, but immunoglobulins don't require the antigen to be associated with the MHC protein. Now let's talk about superantigens. Superantigens are antigens that are not internalized by the cell. They are also larger in size than the normal antigens. The normal antigens are internalized by the cell and then they are represented on the surface by the MHC protein, and then they are presented to the T cells. But superantigens, they directly go and attach to the T cell receptor, and they don't bind with the receptor binding or antigen binding cleft, that is the part of the TCR on which the antigen binds, but they bind to some other region on the receptor. There is also another characteristic of the superantigen, that is they activate a number of T cells, but a normal antigen binds to a single T cell or T cell receptor and activates a single T cell, but the superantigen binds to different T cells at a time and it activates different T cells at a same time. And when those T cells are activated, they produce interleukin 2, and which activates other T cells, which include the helper T cells and the cytotoxic T cells. There are different types of examples of the superantigens. Some of the most important includes the enterotoxins of the Staphylococcus and the toxic shock syndrome toxins. Allah Hafiz.